Hello everybody, thanks for coming. My name is Philipp. You can see from my clothes that I'm a scientist. Regular clothes. I'm a postdoc in Dave Edwards' lab who sits there in the back somewhere in Western Australia, in Perth. But I'm actually German. So today I'll talk to you about what we are doing with NetMiner in relation to plant breeding, because that's our main job. We do Brassica napus canola plant breeding. So as an overview, first I'll talk a bit about what makes plant genomics hard, the challenges we have with Brassica napus and canola. Then I'll talk a bit what we've been doing with NetMiner so far. And then the rest of the talk I'll spend, as usual in my talks, I'll spend complaining about stuff and what we're going to do soon, and all the bad things, and what's going to happen in the future with NetMiner. So the challenges in plant genomes we have is that, who in here works with plants? I'm pretty sure most people do, isn't it? Some hands. Half-ish, yeah, okay, that's good. So plant genomes are usually huge. They are repetitive. They have many copies. You have many homologous copies inside the same, so you have the same gene a few times inside the same genome. And often the genes we personally in our group are interested in are extremely repetitive too. So we are often working with resistance genes, and you'll have always like 5, 6, 7, 8 copies of those. And that makes bioinformatics analysis relatively hard. This is the main thing we work with. This is the triangular view. Some of you have probably seen it 1,000 times, the people who work in Brassicas. If you've been to some of the Brassica talks here, you've seen it 5 times, 6 times. So this is, for those that haven't seen it before, this is the triangular view named after a Korean-Japanese scientist from the 30s. He showed back then that Brassica, rapa, Brassica nigra, and Brassica oleracea are actually related. They all come from the same ancestral genome. And you can merge these together. So Brassica napus canola is the one we're working with, is an AC genome. So these two genomes merge together. It's a fairly recent event that happened. There have been a few hybridization events. We're still fighting a little bit when that exactly happened. Some people say a few hundred years, some people say a few thousand years. It depends what you ask. And of course, since these genomes come from the same ancestral genome, they overlap largely. So A1 and C1 are relatively similar in the context, in the genetic context and in their structure. Then again, that makes it harder, and that's why knowledge networks are so useful in this case. So, people have been asking us bioinformatics, people now for 10, 20 years, what's it good for? What's all this bioinformatics stuff good for? Now the plant readers say, well, you have a genome reference, and you have the annotation, and you have this knowledge network, what is that good for? This is a nice example from Bayer. I got the slide from Bart Lambert, thankfully, thank you, showing actually a nice approach of where bioinformatics was used in plant breeding. In this case, it's a new plant called PodGuard, a new plant cultivar for Brassica napus. There is a known shattering gene, so the seed is in these pods. The seed of the plant is in these pods, and when you usually harvest them earlier so that you can just take directly the pod, and then you shatter the pod in the factory or something and pick up the seeds from there. What happens often in the field with Brassica napus, with canola growing, is that these pods open too early, and then the seeds fall on the ground, and since they're really tiny, nobody bothers picking them up, so you lose yield. You lose like 10% of a yield or so to that phenomena. So people usually harvest earlier so that the pods don't open. And then, of course, you lose yield again because you harvest also the plants that haven't matured yet. So we have a known shattering gene that is linked to the opening of the pod, and we have 2 homologs in the Brassica napus genome sequence that came out 2 years ago, but has been available a little bit before that too. So there's a homolog on A3, and it's equivalent on C3. And the Bayer people showed with their transcriptome atlas that this gene is only expressed in the pod valves. So it's where it should be. You don't have any off-target in the stem or in the flowers or whatever. So they first made a GM plant. They made actually 2 different GM plants based on that reference gene where they knocked it out. And that one worked too well. There you couldn't open the pods at all. Which is, again, that's not good. But it's a nice approach to show, look, we use bioinformatics to find these genes. We actually use the knowledge we have in our networks or in our databases to predict how it's going to look like in the actual plant. And they had to go back to the tilling population and found a few individuals that have that show variety in their gene. So they use those as parents for novel plants. And you can buy this plant not this year. I think they've been starting to. The farmers have this plant this year in Australia. and it's got about 10% more yield than equivalent plants, which is, well, that's a good success story for bioinformatics where it's actually helping in plant breeding. And this is also where NetMiner comes in. So we're starting to put all this breeding information we have into NetMiner instances. It's a lot of heterogeneous, a lot of complex information. And based on that nice network, then the lab people, the non-bioinformatics people too, can go in and look for their genes of interest. They can look for the information of interest and decide from there how to go. So how I got involved is I'm a German citizen, as you can hear from my wonderful accent. And I had a bit of a visa mishap, which probably some of you have had before, and I was stuck in Germany waiting for a new visa. And at the time, my boss, David, said, do you want to work with the NetMiner people in Rothamsted Research? The UK isn't far away. From Perth, it's super far away, but from Germany, it's just a 2-hour flight. So I flew over for a week, improved a lot of the documentation, and set up our first Brassica oleracea instance. Back then, I used Brassica oleracea, so only the C genome, which is kale, cabbage, all these vegetables, because it's just a smaller genome and it's easier to set up. But by now, in the last few years, we've set up a few more instances. We are now running a Brassica oleracea with the pan-genome annotation. We're running an instance for Brassica napus with a few different annotations from different cultivars. And we're running one for chickpea, because that's also what we've been involved in the chickpea genome projects. So I'll show a few examples from these instances now. Who saw Dave Edwards' Brassica oleracea pan-genome talk? Did anybody see that? He saw it, of course. Very good. So I'll explain that a little bit. What we did is the reference assembly we have for Brassica oleracea doesn't have all the genes that are in all the cultivars of Brassica oleracea, of course, because the reference assembly just shows the state of one single cultivar. It doesn't have what's there in the whole population. And we have a paper out a few weeks ago in Nature Communications where we looked for genes in other Brassica oleracea cultivars that are not in the reference, and added those. And we found a few thousand of those, which are because Brassica oleracea has such a huge morphological variety and such a diverse history, so you find many, many genes that are not in the reference. And they are in this graph. There are these genes called BOLEPAN, Brassica oleracea pan-genome. This gene here is just that's the main reference gene, BO6, it's on chromosome 6, and there's a few Arabidopsis genes up here. Proteins, sorry, these are Arabidopsis proteins, and that's a gene here. And these red bars are blast overlaps. So it's not shown here, but these blast overlaps are not very good, which makes sense. It's a very different gene from that one. This here is a resistance gene. I just wanted to find a few resistance genes for my project. So we found this resistance gene, which is related to the resistance genes in Arabidopsis. And we know that the novel pan-genome proteins we have, they're slightly related to this one. So there could be resistance genes. We're still checking that. So that's my example for Brassica oleracea. This is a bigger example from Brassica napus, from canola. Down here is the Brassica napus gene. It encodes for a protein, and this is related to an Arabidopsis protein, and there's this huge body of scientific literature on that gene. These are all resistance papers. Do we have water? Oh, sorry about that. Yeah. So it's not shown in this graph, but there's a few known SNPs in this gene. And from there, we can then go to the plant breeders, to the companies, and ask them, look here, we got 4 SNPs of interest in our gene of interest. Can you please check your breeding populations, your mapping populations, if you see any variety in these SNPs? And from there, maybe use these as parents for novel cultivars. And lastly, this is an example from chickpea. So we have a chickpea genome here, which is again related to a flowering time gene. I think K1 stock had ELF3 before. This is ELF5, so it's fairly similar. So yeah, that's a novel gene too. We didn't know about that one before. So all my examples today are more about homology to Arabidopsis and to other information resources. But we also have SNPs and domains and trait ontology and everything linked to these genes too. But this is just currently what we are mostly interested in, to find novel genes nobody knew about before. So now comes the part where I complain, because I'm a bioinformatics person, I always complain. These are the 4 things I'm going to complain about. The first one is that the Arabidopsis, the data sets you have, the best, most well annotated organism in plants is Arabidopsis. But Arabidopsis is such a tiny genome and it doesn't have many of the resistance genes we are interested in, of course. So it's starting to get limiting that everybody makes all this Arabidopsis data. And there's 3 more. I'll talk about them on their own slides. So these are the top 70 organisms in CRISPR. So by the number of their proteins, of the genes annotated there. Everything in bold is a plant. So we have one up here, of course, Arabidopsis. We have rice, and all the way down here is rice again, and a bit of maize. That's it. The rest is bacteria, boring humans, diploid stuff. You don't need that. But yeah, to show that CRISPR is a wonderful database. It's so useful, but it's mostly Arabidopsis. And again, for us, that's not the most useful thing. That's something KnetMiner, of course, cannot fix. That's something the community fixes because people know that genomes are getting so cheap and so easier to generate. People are starting to move into actually useful organisms. But I have to also say that KnetMiner also has the plant network from, what's it called again? What? Come again? The whole plant network. It's not just Arabidopsis, of course, in the networks.
Yeah, it's got the reviewed proteins from UniProt can be CRISPR in there as well.
Okay, so all of CRISPR is in there. The other thing I wanted to complain about, which we started to discuss, we're starting to do, is we have many lab people who don't do much with bioinformatics, who don't do much with computers. They usually run their Galaxy instance or something like that. So they pop their gene of interest in there and run a small alignment, and that's it. What I would love to do, what some people have started to do, is I would love for that lab person to come, take their protein or gene of interest, put it into KnetMiner, and that one then checks out the next neighbors in the graph and generates a plain English 3, 4, 5 sentence summary on that. Maybe we show that next year. I'm not sure. That would be very useful. It's kind of your text mining, but reverse.
Yeah, reverse.
Yeah.
Right. The English might be very plain.
Yeah. Which, yeah, that's okay. And of course, that's another problem we have is it's so cheap and easy to generate genomic data. It's still expensive to generate phenotypic data. It's still expensive to look for pangenomes. It's still expensive to generate genetic variation data, association data. I have so many reads on my work server, but I have very little phenotypes. And that's something, again, the community has to work on more. And yeah, we do that more and more now. And there were some nice talks on automated phenotyping too, very interesting. And the last thing I wanted to complain about, that's another thing we've been working on, is to not only give a score to the genes, but also to the evidence itself. So by looking at how rare is the evidence in the entire network compared to the network of results we have. There's an open issue on GitHub to implement that, which has been assigned to a specific person in this room quite a while ago, and that specific person has done something, but then that specific person had to finish his thesis. It was me. So it would have been in there with all my thesis. Yeah, that's something that I'll do that once I come back to Australia, if they let me keep my visa. So, and the next thing is, you've seen this in a ton of talks here at PAG as well. CRISPR crops, there have been so many examples from people who use CRISPR to kick out a piece of the genome or to insert a promoter or something like that to make a new cultivar. Monsanto just got a license to run CRISPR from the Broad Institute, I think. Yeah, from the Broad. So they'll do that more and more now. Here's a recent example from a publication, but I don't know. I think everybody in this room has seen a CRISPR example in this conference now. This is just an early yield variety from tomato. You can see that here, instead of growing, you get, at the same time, you get already finished ripe tomatoes down here too. You don't waste the energy on growing up high, you just start to flower down here and get your tomatoes earlier and quicker, which is very useful. So, yeah, it feels like there have been 1,000 talks on this in PAG. So KnetMiner is, I think, an ideal approach to find candidate genes for that. Like I want to, for example, I want to change something in relation to resistance, so I go into the graph, I look for my novel genes, especially if you work in an organism that isn't so well annotated, like chickpea, for example. And from there you can then find the candidate genes or candidate areas in the genome for CRISPR manipulation. But yeah, there's lots of work to be done yet. We just started to get into CRISPR ourselves, so maybe PAG 2018 there will be a nice talk. And that's about it. These are the acknowledgements. You can contact either Dave Edwards down there. I can show the pointer at him. Or me, or on Twitter. That's me too. I have to thank Rothamsted, of course, the team of Rothamsted, Ajit for always answering my weird emails at 3 a.m. asking about stuff. Dave and Jackie. Jackie isn't here, is she? Nah, I would have pointed her out. And that's our team. And I also have to thank Biocrop Science. I didn't put their logo there, but Biocrop Science gave us a few slides and nice examples for this talk. So if you have any questions, I'd be happy to answer them, or maybe not. No, I will answer your questions. Ah, yeah, that's my last slide. That's my son. He doesn't have anything to do with this talk, but I like him very much, and I'm very proud of him. And I've shown him to everybody. I know you saw him too. So yeah, that's a Any questions? We have a lot of time.
I do have a question.
Yes.
So you mentioned that currently you want more than just arietinum,
Yeah.
and that other plant species are underrepresented in CRISPR.
Yeah.
Is this an issue of that data not existing out there, or is it in other repositories that haven't yet been integrated with this tool?
Ah, I see. I don't think it's out there yet, especially not in the repositories. I mean, the Brassica napus annotation is just 2 years old, the public one, and nobody has gone through and manually checked which one, what's actually good here. And I did a few. Yeah, it's just not out there yet. It's just now starting to get cheap enough to sequence all these nice big plant genomes. I mean, you've probably seen some of the wheat talks too. That you've got many new annotations there from different providers. So these will be in SwissPot in maybe 5 years or so.
And SwissPot, it's manual annotation quality.
Yeah, it's a massive amount of work. Did that answer your question?
Yes, it did.
Good. Any other questions? Or you want to hear the next talk? That's good too. Okay, good. Then thank you. Thank you for looking at my son. I take this.